Welcome to Nittakis. My name is Mrs. Ramla. I have prepared this video to help the grade 12s with their final exam preparation. In this video, I'm going to be looking at the Feb-March 2015, the paper 1. Right. Give the correct biological term for each of the following descriptions. Write on only the term next to the question number in the answer booklet. 121. The structure in the ear that equalizes pressure on either side of the tympanum. Ear, it says eardrum. I want us to get used to the word tympanic membrane or tympanum. We don't accept the word eardrum anymore, but this was 2015. And that we know, whenever you see the word equalizes pressure, that is eustachian tube. 122 and 123 I've removed because it's based on human impact, so that's not part of the syllabus anymore. 124. The watery fluid that supports the cornea and the front of the chamber of the eye. Immediately when they say watery fluid, you must remember that that's aqueous humor. 125. The hormone produced by the Graafian follicle, that we know is estrogen. 126. The hormone responsible for the formation of the corpus luteum, we know is luteinizing hormone. 127. The receptors in the ear that detect changes in the direction and speed of any movement in the body, that is the cristae. Right. 128. A hormone which stimulates the secretion of thyroxine, that we know is thyroid stimulating hormone. And then 129. The type of fertilization associated with viviparous reproduction, that we know it can only be internal fertilization. And the last one, 1 to 10. A series of changes that take place in the shape of the lens and the eyeball in response to the distance of an object. The key word is distance. So distance, we always use the word accommodation. That's what it is. So when the lens and the eyeball changes in response to distance, that's called accommodation. Right. Our matching columns. Indicate whether each of the statements in column 1 applies to A only, B only, both A and B or none of the items in column 2. Write A only, B only, both A and B or none next to the question number. Now can you see here there's 6 of these matching columns. In the past, there used to be a lot of matching columns and the learners never used to follow the instruction. So currently in this year, there's only 3 to help reduce the number of mistakes that the learners can make. 131. What may cause a decrease in the pH of the blood? Now, we know that glucose will not affect blood pH, but yes, excess carbon dioxide will affect blood pH, so your answer is B only. 132. The part of the brain that connects 2 hemispheres together, we know is not the cerebellum, it's the corpus callosum, so your answer is B only. 133. The brain disorder that results in memory loss. Now, memory loss is obviously definitely associated with Alzheimer's disease, but not multiple sclerosis, so your answer is A only. 134. A structure in the nervous system that detects a stimuli. Now, what picks up a stimuli? Now, we know it's not an effector, but it's a receptor, so your answer is B only. 135. A hormone secreted by the pituitary gland or the hypophysis. Now, testosterone is secreted by the testes and thyroxine is secreted by the thyroid gland. So it is none of them. So your answer, none. And the last one, 136. A type of development in birds in which the young are capable of moving around soon after hatching. We know that's precocial and not altricial. So our answer is A only. Make sure here you follow your instructions. You write either A only, B only, both A and B or none. Right. 1.4. Study the diagram below, which shows a process occurring in the human male. 141. Name the process by which male gametes in humans are formed through meiosis. That we know is spermatogenesis. 141. Name the organ in the males where spermatogenesis takes place. That's what they're asking. In the testes. 143. How many chromosomes will be found in each cell at A? Well, there we already know it's the end of the meiotic division, so it will be 23. And then B, how many chromosomes? Still 23 because it's second meiotic division and 4 cells are found. Right, 144. Name 2 processes occurring during the first meiotic division that contributes to genetic variation of cells at A. Of course we know it's crossing over and random arrangement of chromosomes. Here the memo says assortment. Let's get used to using the word random arrangement of chromosomes at metaphase 1 or metaphase 2. Then 145. How many cells at B will carry the Y chromosome? We know that answer is only going to be 2. And then 146. What are the mature cells at B called? Easy. Sperm, spermatozoa, or the male gametes was also accepted in the memo. Right. 2.1.1. It says they study the diagram below, and we can see that's the male reproductive system. Give the label for each of the following. A. Can you see A is a coiled structure on the testes, so that can only be the epididymis. Give labels for each of the following. B, of course, is on the testes, so that is obviously the testes. And C, it's the tube that transports sperm, so it's the vas deferens or the sperm duct. 212. State one function of part A. What's the function of part A of the epididymis? Store sperm cells until mature. 213. Explain the consequences for reproduction if part C is surgically cut. Now we know, what does it mean? Sperm will not be able to leave and go to the urethra to fertilize the ovum, and there will be no fertilization, so no children forming. It says the consequence. So last point here in the memo, that's the consequence. Always remember to answer the question. 214. Explain why it would still be possible for a HIV positive man to infect another person during sexual intercourse after part C is surgically cut. Now we know the HIV virus is passed on during sexual intercourse, and it's passed on in the body fluid, meaning it's passed on in the secretions from the accessory glands. So you can still infect his partner. Right. 2.2. Describe how the different parts of the ear and brain allow for hearing to occur. Ear and brain. So what must you say? Sound waves are directed by the pinna through the auditory canal to the tympanic membrane, causing it to vibrate. The vibrations of the tympanic membrane are transferred to the ossicles in the middle ear, which eventually cause the oval window to vibrate. This sets up pressure waves in the perilymph. Pressure waves are then transferred to the endolymph in the cochlea, which stimulates the organ of Corti in the cochlea, which converts this stimulus into an impulse, which is then transported along the auditory nerve to the cerebrum where sound is interpreted. Can you see the last 3 points is in bold? That's the compulsory marks. That means you had to answer this question. If you didn't get the compulsory marks. So make sure when you read the question, you answer what they're asking you in the question. 2.3.1. So before we look at that, let's say study the diagram of a reflex arc below. 231. What is a reflex action? We know a reflex action is a rapid automatic response to a stimulus. 232. Label the following, the functional connection. So immediately when they say functional connection at D, I know that answer is a synapse. Neuron B, that's easy. Can you see it's in the spinal cord, in the gray matter? So it's the connector on the interneuron. 233. State the significance of the functional connection at D. They're asking you about the synapse. What's the significance? Ensures the impulse travels in one direction only, prevents continuous stimulation of the neurons, and ensures that the impulse is transmitted from the sensory neuron to the motor neuron. This is a nice answer. Write it down in your books. 234. Write down in the correct order the letters only of the neurons involved from the time a stimulus is received until a response takes place. So this is the receptor. So it will be A, then neuron B, and then of course C. So ABC, that's how you write it down. 235. Explain the consequences for reflex action if neuron C, which is the motor neuron, is damaged. The person will be able to receive the stimulus but will not be able to respond, right? And so there'll be no reflex action, there'll be harm to the organism. And 236, they want you for 236 to draw a label diagram to represent the structure of A. They want you to draw a sensory neuron. Now this is how you draw a sensory neuron, very important, with the cytoplasm, the cell body, and the nucleus, axon, dendrite, don't forget myelin sheath, and then don't, very important, have the direction of the impulse. Let's put that here, direction of impulse. so you can see. Right, so that's very important. Make sure you have that. This was a very nice question, grade 12s. A very nice question. And a lot of different ways they can ask this question. So I really like this question. Right, 2.4. The graph below shows the blood glucose concentration in a normal person and in a person with diabetes mellitus. Both persons ingested 100 ml of glucose solution at 30 minutes, as you can see there in the graph. Right. So what does the graph show us? Blood glucose concentration in grams per centimeters cubed during 2-hour period. You have your blood glucose concentration in grams per centimeters cubed over time in minutes. 241. On this graph, before we carry on, it's the person with diabetes mellitus and a normal person. So now, 241. What is the blood glucose concentration in grams per centimeters cubed of a person with diabetes mellitus at 90 minutes? So where's 90 minutes? There's the person with diabetes mellitus, and there we read our answer, 0.25 grams per centimeters cubed. 242. How many minutes after ingestion of glucose did the glucose concentration reach its highest level in the normal person? So after ingestion. So it reached its, there's it there, after ingestion, reached its highest. That 45 minus 30, 15 minutes. 243. Describe two differences in the pattern of blood glucose concentration with a person with diabetes mellitus and a normal person for 2 marks. Now this is very nice answers. The blood glucose level of a person with diabetes mellitus, can you see it's much higher than that of a normal person all the time. There's a greater increase in the blood glucose level of a person with diabetes mellitus after ingestion compared to the normal person. And it takes longer for a person with diabetes mellitus for their blood glucose level to stabilize compared to a normal person. I like this question, especially if you look at the memo, they want certain things. So make sure you practice this question and get used to how they want to answer, how they want you to answer in the exams. 244. Explain the reason for the differences mentioned in 243. Now, the person with diabetes mellitus does not produce insulin or is insulin resistant. That is why the blood glucose is not converted into glycogen. And 245. Name two hormones that have the opposite effect to that of insulin, glucagon and adrenaline. Right. 3.1. Study the flow diagram below of a homeostatic mechanism used to regulate the concentration of salts in the human body. Right, so you have the receptors in organ A, you have decrease in salt, increase in salt. Then you have gland B starting to secrete hormone C, gland B stopping to secrete hormone C. Then you have effector response D, effector response E, and normal salt concentration. 311. Define homeostasis. Now, what is homeostasis? The process whereby the human body is able to maintain a constant internal environment. 312. Give the name of organ A. Where are the receptors to pick up sodium or salt? That is obviously in the kidney. Give the name of gland B. Oh, obviously, the receptor picks up the amount of sodium and gland B will start secreting the hormone, which is the adrenal gland. And what is hormone C? The adrenal gland will start secreting aldosterone. 313. Describe the response by effector at D. Now this is when there's a decrease in the salt or sodium concentration. So what will happen? The walls of the renal tubule will become more permeable to sodium, so allowing for greater reabsorption of the sodium ions from the filtrate, and that obviously will bring the sodium levels back to normal. Right, 3.2 and 3.3 I've removed from the paper because it's human impact, and human impact is not in our exams anymore. Right, 3.4. Tobeka investigated the effect of auxins on the growth of 3 plant shoots, A, B, and C. The plant shoots were treated as follow. Can you see A? No, treated in any way. B, can you see? The tip is removed and an agar plate with auxin is placed on top. Shoot C, the tip is removed and an agar plate without auxin is placed on the top. All shoots were exposed to the same light conditions. Note, agar is a jelly-like substance that allows auxin to diffuse through it. The diagram below illustrates the setup at the beginning of the investigation. 341. Identify the independent variable in this investigation. So what are you choosing to what are you choosing to control? Right, you are choosing the treatment of the plant, shoot, as you can see from the aim that's written here. Then 3.4.2, state 2 factors that must be kept constant in this investigation. The same type of plant, I would not have said type of plant, I would have said same species of plant. And the plant must be placed in the same environment, the plant must be placed for the same amount of time, the tip must be removed at the same length, they must have the same concentration of auxin and the same type of agar. 3.4.3, explain the results observed in shoot B after a few days. Now, what will happen? Shoot B will grow upwards, right? Why? Because the auxin diffused downwards, right, and into the shoot. And as it diffused downwards, what will happen? Cell elongation takes place. Okay, so that's what will happen. What will happen in shoot C after a few days? Now, there will be no growth in shoot C. Why? Because the shoot tip does not contain any auxin, so there cannot be any growth. And then 3.4.2, suggest 2 ways in which Rebecca could have improved the reliability of the investigation. Easy. She could have repeated the investigation and used more than 1 plant per investigation or increased her sample size. Right, the last question. It's the essay question. I still do the essay question because it can still be taken out and asked in small questions in the exam. The unicellular zygote undergoes many developmental changes until it becomes a multicellular fetus, nourished and protected by the mother. Describe the changes that allow the zygote to eventually develop into a fetus and how this fetus is nourished and protected during the period of pregnancy. So, zygote developmental changes. The zygote divides by mitosis to form a ball of cells called the morula. The morula will undergo more mitosis to form a hollow ball of cells called the blastocyst. Right, carry on. The blastocyst attaches to the endometrium lining. The outer wall of the blastocyst is called the chorion, which will develop projections called villi, chorionic villi, and will implant in the endometrium lining. Here it says wall, I don't agree, it must be lining. Then the cells of the embryo continue to divide and differentiate and form different organs and limbs, and this is now called a fetus. The fetus is enclosed in a sac called the amnion, filled with amniotic fluid, which protects the fetus against temperature regulations or fluctuations, which protects the fetus against dehydration, and protects the fetus against mechanical injury by acting as a shock absorber. Then lastly, the chorionic villi and the endometrium form the placenta, where the blood of both the fetus and the mother run close to each other, allowing for nutrients to diffuse into the blood of the fetus. The umbilical vein will carry the absorbed nutrients from the mother to the fetus. This is a very nice question, grade 12s. I would write most of this down as this question is making a comeback now in these days. Right, grade 12s. Thank you for watching this video. I hope you learned a lot. Join me and watch all my other videos as well.